Now that we understood what's going on in the general sense of the light reaction, I want to explain what exactly happens and what you need to remember for the IMAT and BMAT exam. So this is photosystem 2. In photosystem 2, something very interesting happens. You have the oxidation of oxygen, of water molecule. And it's very, very rare in nature to actually cause the oxidation of water molecule, because water molecule is usually causing the oxidation of things. It doesn't being oxidized by itself. So you have the removal of hydrogen and electrons from water, and you cause basically the formation of oxygen. How do we do it? So light comes to photosynthesis too, and it's going to jump from simple complexes to another. They are jumping between one to another all the way until they reach this P680 spatial pair. So the photons come and jump from different proteins all the way until they come into the active zone. In this active zone, which is called the P680, we are basically going to have the electrons now waiting. The electrons are waiting because we split the water molecule, we took the electrons, and now we have electron over here. The electron is going to wait until photon is going to kick the electrons and cause it to be in the excited state. Once we kick this electron using the photons, the energy from the sun, we are going to reach a complex that is basically going to catch the electron that the photons basically kicked out and kicked up and got excited. Now the electron is in the excited state. It is going to go through one complex to another until we reach the second photocomplex, photosystem. Now we are going to have the electrons again. This electron is going to be in the P700 spatial pair. But it wasn't from water here. We don't have any splittage of water in this photosystem 1. We only have it in photosystem 2. It's the same electron from the first water molecule we split. This time we have the photons jump from one protein to another inside this complex, which you are not really need to remember the names of the proteins, until it come here and again kick up the electron again, charge it again. Now in this state we are going to have something very special. After this part we are going to have a spatial electron transport chain, which is going to help us reach the NADP plus reductase enzyme, and basically we are going to charge and create NADPH using a proton and the NADP plus that we got and we used in the Kelvin cycle, which we are going to see in the next video. So let's sum this video up and understand exactly what's happened in the electron transport chain and the light reactions. Light is come and jumps from different places all the way until they reach the active zone of photosystem 2. The photosystem 2 contains one electron, which we got from the splittage of water into protons and electrons. The electrons are going to jump, the electron is going to jump into his excited state, and then it is going to jump from one different protein to another all the way until it reaches the second photosystem complex, which is photosystem 1. Along the way, we were able to pump protons to the lumen itself, because this is what we use the electron for, in order to charge the pumps, in order to allow the electrochemical gradient, very similar to the mitochondria. Now we are in this complex. Again, we have light, which is jumping between different complexes all the way to the active site. We have the same electron from the first one going to the active state, and now this time we are going to reduce NADP+. While we are doing it, as you can see, we are pumping a lot of protons against the concentration gradient similar to the mitochondria, and we reach a very rich lumen with protons. Now the ATP synthase, which is very similar to what we have in the mitochondrial ATP synthase, is basically going to allow the passage of the protons back to the stroma. And now we have the ATP synthase working, and as we know already, ADP plus phosphate, ADP plus phosphate is going to give us ATP. And this entire process is the light reactions, the formation of NADPH, which is the reduced form and the active form that we can actually yield energy from, and ATP, which is allow us to create energy and allow this energy to go and to be used in the Kelvin cycle. I hope this video made sense. In the next video, we are going to see how we use ATP and NADH when it comes to the formation of sugar and when it comes to actual autotrophs and plant cells.